subjects, which is why we send them the slides so they can take a look at some of the language. But you know, I was rewatching the recording we did with Josh a couple months ago, and like Josh's stuff is pretty technical, and they did pretty well. But they're trying to mumble. Yeah. I mean, the thing that's a little frustrating is like cold too. The thing that's a little frustrating is like we haven't gotten any indication that we need be 508 compliant. Like no one said I'm, you know, I decided to handicap like hearing handicap in our land text. It's just we're required to make everything public. Yeah.
So yeah, we go.
Oops.
We go.
All right, good morning, everybody. Hope you're doing well. Welcome to EPA's Computational Toxicology Communities of Practice. My name is Cameron Clark. I'm a student contractor here at NCCT. Today we're going to be hearing from Dr. Derik Haggard, an ORISE postdoc in NCCT. And he's going to be presenting on the High-Throughput H295R Steroidogenesis Assay and its utility as an alternative and a statistical approach to characterize effects on steroidogenesis. So if you have questions, hopefully we'll have a few minutes at the end to get to those. Feel free to type them into the chat box if you think of them as he's presenting. And if you have any issues with audio or seeing the slides, just speak up and type into the chat box and let us know. But with that, I will let Derik take over.
All right, thanks, Cameron. Thanks for giving me the opportunity to talk about the work that we just published recently since I joined NCCT, and thank you all for your attention this morning. I'll try and keep this brief because our holiday party starts in about half an hour. So I wanted to give a little outline, sort of general view of what I'm going to be talking about today. So I'll give a background on steroidogenesis and sort of why we as a center care about steroidogenesis overall, give the two primary objectives for the research, and then give a background into the high-throughput H295R assay that we use for this study, and then go over the methods and results for the two primary objectives, and then finish it off with some summary and conclusions. So proper steroid hormone biosynthesis and metabolism is essential for a wide variety of physiological processes. In utero, it plays really important roles in fetal development, and in adults, it plays an important role in reproductive function. And for both of those, the sex steroids, so your estrogens and androgens, really play a role in those two functions. But we also know that disruption can result in a wide variety of other adverse effects. For example, congenital adrenal hyperplasia, which presents with that disorder as in and of itself presents with a wide variety of different symptoms and adversity based on what steroid enzyme in the pathway is disrupted. And disruption is also associated with sterility, prenatal virilization, salt wasting, hypertension. So the proper ability to synthesize steroid hormones is really critical. So over 90% of steroidogenesis does occur in the gonads. So for males, that's primarily in your Leydig cells, where vast majority of steroid is testosterone that's being produced, and then for females, it's in the follicular cells with the estrogens. However, the adrenal gland is also an important source for steroid hormones, primarily the corticosteroids, so aldosterone as well as cortisol are the main corticosteroids produced by the adrenal glands. And there are four major classes of steroid hormones, and I've given examples for four. So progesterone is a prototypical progestogen, as the name would suggest. Cortisol is an example of a corticosteroid, and then obviously you have testosterone for your androgen, and then estradiol for your typical estrogen. So we care about steroidogenesis and the proper function of that, and the EDFP was tasked to screen chemicals for endocrine activity, as we all know, primarily focusing on estrogen, androgen, and thyroid hormone signaling. And this program developed an initial tiered screening approach for examining potential endocrine bioactivity of compounds, and that really relied on a panel of low-throughput assays. And so as part of the modernization of the endocrine disruptor screening program to sort of face the challenges of 21st century toxicity, there's been work to start incorporating high-throughput and computational approaches to begin prioritizing just the vast universe of chemicals that fall under examination for the endocrine disruptor screening program. And as I previously mentioned, altering hormone levels through disruption of the biosynthesis of these steroid hormones, as well as how these steroid hormones, once they're produced, are metabolized, obviously can contribute to endocrine disruption. And primarily, this has been relatively difficult to assay in vitro. A lot of these steroid hormones are being synthesized in different organs throughout the body, so finding in vitro models has been a challenge. However, within the endocrine disruptor screening program, there is a current Tier 1 assay that's based on the OECD-validated H295R Steroidogenesis Assay. And this was established for the OECD and then adopted by the EPA, and it uses H295R cells to detect potential perturbations of estradiol and testosterone synthesis. However, like many of the other tiered screening approaches for the EDSP, it's relatively low throughput. So our first objective for this study was to compare this test guideline data to the recently developed high-throughput H295R assay, which I'll describe briefly. And I'll primarily refer to the work by Carmouse et al. In 2016, where they sort of benchmarked and looked at the overall performance of that assay and comparing that high-throughput version to that test guideline. And then I'll tell you a little bit later in the next couple of slides, but the high-throughput assay does include measurement of 13 total hormones, which gives us a sort of a snapshot of the overall effects of compounds on the steroidogenesis pathway as a whole. And so our second objective was to develop some form of a summary measure that allows us to integrate data from these 13 hormone measures that gives us an overall quantification of pathway perturbations. One that gives us a ranking metric that we can then use to prioritize compounds that have large effects on the overall pathway for evaluation in later screening. So a little background on the high-throughput H295R assay that we used here. This was initially a result of a contract with Ciprotex and Opans, and this assay uses the H295R cells, which are a form of adrenocortical carcinoma cells. And these cells are unique compared to other steroidogenic cell lines because all of the major biosynthetic enzymes for the whole steroidogenesis pathway are present and active and functional within these cells. And that's sort of shown by the cartoon here, where in black we have the steroid hormones, and then the white bars indicate the steroidogenic enzyme responsible for synthesizing those hormones. And then we have the colored boxes indicate those four major classes of steroids. So we can see that within these cells, they're able to produce progestogens, corticosteroids, androgens, and estrogens. The cell line is also unique because it has characteristics of undifferentiated fetal adrenal cells. So as you can change the culture conditions that these cells are cultured in to stimulate their overall production of steroid hormones in general, or you could co-culture them with specific factors that make these cells produce more aldosterone versus cortisol or vice versa. So there's some flexibility with the types of conditions and output that you can get from these cells. And for this assay, since these cells capture the whole steroidogenesis pathway, what we measure is the production of 13 of these 13 hormones and intermediates, and we use that using HPLC tandem MS. So just a brief overview for the assay method. For the concentration response study, we did, Carmouse et al. Did do single screening and concentration response, but I'm going to not talk about that because the data that we focused on for this study was our concentration response. So the cells are plated overnight, and then they're they're given a pretreatment of pre-treatment of 10 micromolar forskolin, and that stimulates steroidogenesis through adenylyl cyclase activation. And so we sort of ready these cells and increase their overall steroidogenic output. After 48 hours of that pre-stimulation, we then add chemical or our DMSO controls, and we use a 6-point concentration curve for those chemical exposures, and then they're exposed for 48 hours. And then after 48 hours, the media is removed, and the cells are then tested for viability using the MTT assay. And then the media that was removed from those cells is then sent to op-ands for quantification of those 13 steroid hormones using HPLC tandem MS. So moving on to the first objective of the study, where the primary question was, does the high-throughput H295R assay replicate results from the OECD test guideline assay? And in order to do this comparison, we're using reference chemical data that was acquired when the OECD assay was validated, and that was taken from the study by Haggard et al. In 2011. And so the major differences between the high-throughput form of the assay that we've done here and the OECD reference chemical, some of the major differences is the number of chemicals that were tested for those two screens. So for the OECD guideline, they screened 28 chemicals in multiple concentration, whereas for our high-throughput screen, we screened a total of 656 chemicals at multiple concentrations. The cell culture conditions are different between the two assay designs. And I guess the major difference is in the OECD test guidelines, the cells are not pre-stimulated with forskolin. So when the chemical is added, they're still at their basal steroid production versus the high throughput where their steroid production has been sort of amped up through forskolin.
Excuse me, the slides are no longer being shared.
All right, let's take a look here. Sorry about that. Just bear with us for 1 minute. All right, let's see what we got here. All right, so can folks on the line see the slides now? See the screen being shared now? You right here?
Oh, it's right there.
Yeah.
All right, excellent. Sorry about that, and thank you for speaking up.
My view's all strange. Hold on 1 second. It's weird.
Is it cutting off somewhere?
Oh, no, it's not. Okay, it's because the taskbar is sort of hiding it. All right, so hopefully you guys didn't miss too much. Oh, is that what it is? Oh, there we go. Okay. All right. So I guess one of the primary differences between the test guideline assay and the high-throughput version is the total number of steroid hormones that are measured between the two assays. So for the OECD test guideline, as I've already mentioned, they only measure 17-beta estradiol or testosterone production in these cells, whereas for our high-throughput form of the assay, we're measuring a total of 13 steroid hormones or hormone intermediates. So that's really the primary difference, is the breadth of the pathway that's covered in the high-throughput version. And there it goes. Oh, wow, there's a delay. Did we lose it again?
I'm not sure. It's definitely giving us some trouble here.
Connection connection.
Activity loss.
Yeah.
All right. So sorry about that, folks. I don't know if it was an internet issue or what, but the Adobe Connect just closed down on us. So if you'll be patient for just a couple minutes, we'll get it queued up again. Bear with us 1 minute more, please. We're connecting to the hard line. We should be set up here shortly. Thank you. Hello. So we're back in the room here now. Hopefully y'all can hear me. And for those of you who asked, you should not have had to sign out and sign back in. We're just going to see if we can get things queued back up again. I think we're having trouble with our Wi-Fi. So bear with us. Hopefully it stays on course the rest of the way. And thank you, Derik, for being patient with us.
No worries. And don't, speak up if the slides disappear, just so we know. Let's see. Okay. So to make the fairest comparison possible, sort of an apples-to-apples comparison between...
Good night. Is it dropping off again?
Did we lose it again?
That time it wasn't.
Unless that was just the recording started.
Can anybody on the line confirm that they can see everything okay, hear us okay? Nothing's being shared. Can't see anything. Okay, so it dropped off that again. All right, how about now? All right, great. Thank y'all.
Okay. All right, so to make the fairest comparison between the two studies, we wanted to analyze the data in the same way that was analyzed for the OECD test guideline. And so for that, we used an ANOVA with the Dunnett's post hoc multiple comparison procedure with the alpha set to 0.05, and then we used the DMSO control data from the same plate as the test chemicals for that comparison, and we used the same criteria for a positive hit call that was used in the validation study, where 2 consecutive concentrations had to produce a significant increase or a decrease compared to control, or
Sorry, you set off a little fear in us there every time I see a message come up now.
Okay. So 2 consecutive concentrations had to have a significant effect, or we had to observe a significant effect at the maximum concentration that maintained the cell viability threshold that we set. And then we also added a 1.5-fold change from DMSO for better contextual, for better context for the effects that we observe. So when we constructed the confusion matrices to look at the comparisons, the first thing we needed to look at was how many of those 28 reference chemicals in the OECD test validation study were actually screened in our high-throughput screen. And we found that 10 of the 12 core reference chemicals were shared between the 2 studies. Now, a core reference chemical was a compound that was tested in 5 labs during the inter-laboratory validation portion of the OECD guideline study, and 15 of the 16 supplemental reference chemicals were shared between the 2. So really good coverage for the OECD reference chemical set. And we also added a criteria for the OECD interlaboratory results. If we observed equivocal results, where for the core reference chemicals, if more than 2 of the 5 labs failed to report a lowest observable effect concentration, or if 1 of the 2 labs failed to report a low for the supplemental reference chemicals, those weren't considered in our analysis. So we have here is sort of the results of the confusion matrix, looking at the sensitivity, specificity, and the accuracy of the table up top, and then below is sort of a visualization of that. And in this case, we analyzed the data based on whether we observed significant increases or decreases separately for either testosterone or estradiol. And I just want to focus on the revised, the accuracy values here, which is a good measure of concordance between positive hit calls in our data and positive hit calls in the reference guideline study. And we showed that for increases in testosterone, we had an accuracy of 0.9. For decreases in testosterone, an accuracy of 0.82. And then for estradiol up or down, we had accuracies of 0.8 and 0.95. Now, if we compare those accuracy values that we had in our high-throughput data to just the interlaboratory portion of the OECD guideline, we see very similar concordance between the two, where for the interlaboratory validation for any effect on testosterone, the average concordance among the labs was 0.8, 0.88, 0.91, and 0.9 if they were looking at the 12 core reference chemicals versus the supplemental or the entire set as a whole. And similarly for effects on estrogen, they observed interlaboratory concordance of 0.95, 0.84, and 0.89 for the core, supplemental, or the entire set of reference chemicals. So if we just compare that to our accuracy, we see very similar concordance between the results that we obtained in our high-throughput form of the assay versus the OECD interlaboratory validation concordance results. So for the second objective is primarily the development of our prioritization metrics using this high-throughput assay. And the main problem is there a way that we can simplify a high-dimensional question of 13 hormone measures into sort of one value that can rank chemicals based on their overall effects on the pathway? So just to remind everyone, for the high-throughput assay, we're measuring a total of 13 hormones. However, in our case, when we actually looked at all of the data, we found that 2 steroid hormones that are measured, so pregnenolone and DHEA, or dehydroepiandrosterone, were often measured below the lower limit of quantitation. So for pregnenolone, 53% of all the data that was collected was below the limit of quantification, and for DHEA, about 70% of all this data were not detected in the sample. And so we excluded those 2 hormones from our analysis. So in total, we're looking at data, response data for 11 of the 13 total hormones that are measured in the assay. So here's an example readout of the 11-dimensional results for prochloraz. Now, prochloraz is a known inhibitor of aromatase, or CYP19A1, as well as CYP17A1, and so it's known to inhibit enzymes that are involved in steroidogenesis. And so what we have here is overall concentration profiles for all 11 of those steroid measures. So there's 11 plots, and then on the x-axis for all of these we have concentration. And note that the y-axis for all of these will be slightly different because that's based on the overall amount of basal steroid that's being produced for in these cells. And overall we see concentration responsive effects for this chemical, where we see estrogen and estradiol decrease, as well as the androgens, androstenedione and testosterone decrease. And we also see cortisol and 11-deoxycortisol go down, whereas we see the progesterone go up, as well as deoxycorticosterone and corticosterone. Now those 3 steroid measures, or those 3 steroids in the pathway, aren't affected by CYP17A1, so we expect those to go up, since this chemical isn't inhibiting the enzyme responsible for the conversion of those steroids. But for the remaining ones, we would expect a decrease, and that's indeed what we see. So as I've already mentioned, we're measuring 11 hormones compared to what's the 2 that are measured for the OECD reference study. So obviously we're going to be getting more information about disruption of estrogen or just androgen overall, because we're measuring 2 other intermediates in those processes. So that gives us a little bit more coverage of those 2 important steroids. And also, since we're looking at an overall snapshot of the pathway as a whole, this gives us, we could gain information regarding putative mechanisms of steroidogenesis disruption. And also, the OECD test guideline doesn't look at the progestogens or the corticosteroids. So we're getting in just additional breadth of the overall pathway. And so if we look at the overall hit rate of chemicals across the 4 major classes of steroids, we see quite a bit of activity across all the chemicals that were screened. And so we have a Venn diagram representation of chemical hits across those 4 quadrants. So there were progestogens, androgens, estrogens, and corticosteroids. And so I just want to highlight just a couple figures from this. So 307 chemicals that were screened had a hit in at least one of those 4 major steroid quadrants, so relatively highly active compounds. But if we wanted to focus on chemicals that just hit progestogens or the corticosteroids, that represents about, so 13, 44, and 10, that's 67 chemicals. Now these chemicals would have been entirely missed in the OECD reference guideline because we're not measuring progestogens or corticosteroids. So this just sort of highlights the usefulness of this assay in terms of getting additional compounds that could have been missed in that Tier 1 screen. So since we're measuring so many hormones and a lot of chemicals, the goal was to sort of integrate all of these data into a single value for each chemical that can estimate the overall magnitude of effect on the steroidogenesis pathway. And so obviously some of the initial challenges that you can think of when trying to develop some form of a metric is we're dealing with multivariate data. So we have 11 hormones measured per chemical. The hormones are measured from the same experimental well, so there could be some independence issues there. And it's known that some of the concentrations of steroid hormones and some of the intermediates are often going to be interdependent because one product from an enzyme is then a substrate for another enzyme for the product, and so there should be some relationships between substrates and products. And so that would likely need to be taken into account. So we wanted to start asking development of a metric starting very simple. So a simple solution would be, well, let's just look at the distance between concentrations compared to a control in this sort of multivariate space to see whether that gives us a good metric, because we would expect that chemicals that are significantly affecting steroid hormones should be further away in space than the DMSO controls. However, we know that there are some limitations when we're using sort of a simple Euclidean distance metrics. So I'll give a small conceptual example. So here we have data for some arbitrary hormone A and hormone B for a specific chemical at 3 different concentrations, so concentration 1, 2, and 3. And then we also have the dashed ellipse indicates the overall joint error distribution of all of the data. And so we can see that since it's not a circle, it's an ellipse, there seems to be some positive covariance between these 2 hormones. However, if we measured the distance of concentration 2 and 3 relative to concentration 1 using just the general Euclidean distance, we would see that these would be the same distance away from concentration 1, even though concentration 3, since it lies further away from that joint error distribution, it's more standard deviations away from concentration 1 than is concentration 2. So in that case, there's actually more effects, a more extreme effect on hormone A than for concentration 2. So this Euclidean distance really isn't accurately taking into consideration the effects of that concentration on the hormone A levels. And so one of the first questions we wanted to understand is, well, is something similar going on within our data? And so we looked at the correlation among the residuals for all of our steroid hormone data. And indeed, we did see that several of the steroid hormones were highly correlated with each other. So what we have here is a clustered correlation plot of all of the pairwise correlations between the residuals of all of the 11 hormone measures. And just to highlight some examples, so for estrone and estradiol, we observed a correlation of 0.75, and for androstenedione and testosterone, correlation of 0.66, and for cortisol and deoxycortisol, we observed correlations of 0.69. So there appears to be high correlation among some of the steroid measures. And so in that case, it appears that a simple Euclidean distance metric isn't really appropriate for these kinds of data because it will either over or underestimate the effects of chemicals on between hormones that are correlated with one another. So we wanted to find a way, if there was a metric that allows us to sort of remove the overall covariance structure of these data, and it turns out that Mahalanobis distance, which is a similar statistic to the Euclidean distance, is able to adjust for covariance, the covariance structure within your data. And so in this case, it adjusts the covariance among the hormone measures. And so if we if we take that conceptual example again, and calculate the distances within the Mahalanobis space, what it does essentially is it scales and rotates the data for hormone A and B in a way that the error distribution is no longer correlated. So it removes that positive covariance. And so now if we recalculate the distances within that Mahalanobis space, we see that concentration 3 is now 4 times further away from concentration 1 as in concentration 2. So this is giving us a more accurate depiction of the overall effects of that concentration of that chemical on that hormone. So when we apply this statistic to our data, what we're doing is we're calculating the Mahalanobis distance for each concentration of a test chemical within an 11-dimensional space where each dimension corresponds to one of our steroid hormone measurements. So in brief, for the method, what we first do is we calculate the hormone fold changes for each test chemical at each concentration compared to the DMSO control. We then need to estimate the overall covariance matrix of the data that allows us to characterize the noise variance and correlation among all of the hormone measures in the data. With those two values known, we then scale the computed Mahalanobis distance based on the total number of hormones measured for each test chemical. So that gives us an overall mean Mahalanobis distance for that chemical. And so to do this, this is the equation for the mean Mahalanobis distance, and I sort of jumped through the Euclidean distance equation really quickly. But those two equations are very similar, and really the only difference here is NH, which corresponds to the total number of hormones that's measured for each chemical, and then the sigma, which is that inverse covariance matrix. So this is how the distances are then accounted for the covariance between the hormone measures. So the covariance matrix, to estimate that, what we do is we fit all of the data to a multivariate linear model using our natural log transformed hormone concentrations. The matrix of the fit residuals is then used to estimate the variance and covariance matrix. And we had subsets of chemicals that were assayed on different days, and so we considered them as separate blocks. So when we were fitting to the model, we kept the blocks separated. However, when we did our final Mahalanobis distance calculations for the chemicals, we took the unweighted averages across all of those blocks to give us our pooled 11 by 11 covariance matrix, which was used in the Mahalanobis distance calculation. And so in order to rank chemicals using this approach, the maximum mean Mahalanobis distance would be the maximum of the Mahalanobis distance values across concentrations for any given chemical. Excuse me, and we consider this maximum mean Mahalanobis distance, or max MMD, the overall magnitude of effect of the test chemical on the overall steroidogenesis pathway. And so since in general, as the mean Mahalanobis distance generally increases with increasing concentration, that theoretically would mean that a greater max mean Mahalanobis distance should indicate a higher concentration of a test chemical, since for the most part we see a monotonic response in the distances, as well as increased potency, meaning that we observe significant activity at lower concentrations. Another statistic that we calculated was a critical limit, which is, you could sort of think of it as a threshold. So this we derive this value to distinguish the Mahalanobis distance that are greater than what would result from noise or from just random chance, while also accounting for multiple comparisons for comparing each concentration to the control. So I'm going to be giving just two brief examples, and I know because of all the disruption, I'll try and go through this a little bit fast. So example of a very strong effect and then a negative effect from two chemicals. So here we have mifepristone, which is an antagonist for the glucocorticoid receptor, the progesterone receptor, and the androgen receptor. And so we would expect to observe changes in some of the hormone measures, since obviously steroidogenesis involves glucocorticoid and progestogen signaling. And so what I've done is I've condensed that complicated 11 plot figure into a radar plot where each spoke, which is shown on the left, where each spoke is a specific steroid hormone measure, and then each point is colored represents a chemical concentration, where darker colors are higher concentrations compared to lighter. And then also just added the red dashed lines indicate the 1.5-fold change control just for reference. So for mifepristone, we observed very strong dose-dependent increases in progesterone and hydroxyprogesterone, as well as strong decreases in 11D cortisol and cortisol. So relatively strong effects on the pathway. And if we look at the figure on the right, this is the mean Mahalanobis distance plot for this chemical, and I've also annotated that critical limit threshold value. We see a very nice monotonic curve increase in the mean Mahalanobis distance for this chemical, indicating very strong effects on the overall pathway, and also have highlighted the max MMD here. So this value here would be used as our overall ranking value for this chemical. And then the next example is that of a negative. So here we have benfluralin, and again, the same concept where we have the radar plot showing all of the steroid hormone measures. Now note that the y-axis between this and mifepristone is going to be different, which is why the dashed line boundaries of that fold change band is much larger here. But overall, we don't really see any very strong dose-dependent changes in any of the hormone measures, and that's reflected in the mean Mahalanobis distance plot here, where the critical limit in this case is all the way up here, and none of the concentrations elicited a Mahalanobis distance that was above that critical limit, which reflects the overall profile of steroid hormones for this chemical. So since the OECD test guideline really relied on ANOVAs, versus what we're doing here, where we're integrating all these data into some quantitative value that overall signifies the effects on steroidogenesis pathway overall, we wanted to sort of look at if we took that similar approach as the OECD guideline and sort of summarize the data as just a sum of the total number of steroid hormones that are significantly affected by the chemicals. What value are we actually gaining from this max mean Mahalanobis distance metric? And so what we have here is box plots for chemicals that had significant effects on any of the 11 steroid hormones. So 0 indicating that this chemical had no significant effects on any hormones, and 11 indicating that it had significant effects on all 11 hormones in the pathway. And then on the y-axis, we have the max MMD values, which gives us the overall distribution in that max MMD value with increasing steroid hit count. So in general, what we see is as the total number of steroids are significantly affected by the ANOVA, we see a general increase in the distribution of the max MMD, so that sort of makes sense. So, but there's 3 examples I want to highlight here is points that are open circles indicate a negative max MMD response, where the max MMD did not go over that critical limit. So it would be we consider that a pathway negative response. And we actually see our measured pathway negative responses from 0 to 6 significant steroid hit counts. And so this correlates to a significant ANOVA, but very low level activity on the overall pathway as a whole. And so whether that would be a misclassification or not, if we just relied on some of steroid hit counts or not, needs a little bit of exploration, but it's an interesting finding. Another thing to mention is, so bisphenol A was replicated 3 times independently throughout this study, and so that gives us an interesting way to look at the reproducibility of this benchmark of the maximum MD overall. And interestingly, we observed that the 3 replicates had either 5, 6, or 7 significant effects on hormones, but the max mean Mahalanobis distance values were roughly the same for those 3. And when we went and looked back at those data, the steroids that contributed to the 6 and the 7 hit counts were relatively low magnitude changes compared to the primary drivers of this maximum D value, or the primary drivers of the steroid responses within this chemical. And then one last example, I guess is one of the most extreme examples of sort of the quantitative value of this type of metric compared to just some hit count, is for 2 chemicals that significantly affected 6 steroid hormones as measured by the ANOVA. So we have finasteride, which is this red circle, and we just lost it.
Sorry about that. It looks like we got bumped on Adobe Connect again, but we didn't lose internet connection, so hopefully it'll let us pop right back on there. All right.
Okay. So this orange circle represents finasteride, and at the bottom here, the open triangle is EDS, and so this was considered a pathway negative within our maximum MD metric, whereas finasteride was a very strong positive within the metric. But if we relied just on hit count alone, we would sort of consider them as similar responses. However, if you actually looked at the overall steroidogenic profile of these compounds, we see that finasteride largely affected these 6 hormones in much greater than that 1.5-fold change, whereas for EDS, it had very low magnitude effects on the pathway. And so by using this maximum MD approach, we're able to quantitatively differentiate these 2 chemicals as an active and a non-active, whereas relying on just an ANOVA alone wouldn't give us that clarity. So for sake of time, I think I'm going to sort of brush over this really quick. So in the same sense for bisphenol A, there were a total of 107 chemicals that were replicated in more than one subset of chemicals that were assayed with an actual decent range. So this gave us a good way to sort of look at the reproducibility of the metric overall. And if we just looked at the maximum difference between these internally replicated chemicals, we observed a 1.47 median maximum difference between the maximum D values on their arithmetic scale, so relatively stable. And if we looked at the concordance between replicates having a positive pathway response and a negative pathway response, we observed that 88% of our chemicals were concordant with one of the replicated chemicals were concordant with one another. And we contrast that with if we use the ANOVA logic, looking at the concordance between hit calls for those 11 hormone measures. With the ANOVA, we only observe a 65% concordance between the ANOVA. So we're getting an increase in concordance using this overall pathway approach versus simply summing up the significant number of hormones that are perturbed. I think I'll skip over this as we lose it. I should. Okay. So for sake of time, I'll skip this, but just know that when we look at the max MMDs of the OECD reference chemical set, so those 26 chemicals that were tested in the OECD guideline study, we see that the max MMD does a really good job of ranking those chemicals based on their effects on the estradiol and testosterone, where we're able to separate out the strong effects versus the weak or the negative effects using this approach. Okay, so after all the breaks, oh yeah, I'll just go through and talk through these two slides. So to sum up, when we evaluated the high throughput screen to the OECD test guideline, we saw a relatively good concordance between the two, where both our hit call data and the interlaboratory validation both were approaching that 90%. So good concordance between the two studies, demonstrating that the high throughput assay is performing just as well at measuring estradiol and testosterone perturbations as the OECD test guideline. And last one, there we go. Okay, and then by using the Mahalanobis distance statistical metric, that was able to reduce that 11-dimensional question down to a single dimension. And by using the maximum mean Mahalanobis distance for a chemical across concentration, that appeared to provide a reproducible and quantitative approximation of the overall effect of a chemical on the steroidogenesis pathway overall. And this allowed us to distinguish weak, moderate, and very strong modulators of steroidogenesis. And you know, with this type of approach, we get mean Mahalanobis distances across concentrations. We get that maximum mean value. We can model these data based on that critical limit value to give us that lowest concentration that corresponds to significant perturbation on the steroidogenesis pathway overall. So any one of these metrics could be used as some form of rank to review effects on specific hormones, or it could also be used as sort of a lowest observable effect concentration for risk assessment or in some form of context like that. And so, yeah, so with that, I'd like to thank Katie Paul Friedman, my mentor, and all of the collaborators on this project, so Woody Setzer, Richard Jepson, Agnes, and Matt, as well as the ORISE program for giving me the opportunity to work here at NCCT, and of course everyone at the center. So with that, I can take any questions.
Yeah, thank you so much, Derik, for presenting and for bearing with our technical problems, and thank you everybody on the line and in the room for putting up with these issues as well. Hopefully we'll have that sorted out in time for our next webinar. And I want to note that we will have Derik's slides posted on our website. That link is included in the invite. If you have any follow-up questions for Derik that we don't get a chance to address, feel free to respond to that invite, and I will share those with him. So it looks like we have a question on the chat here. Do you have insights into the unique chemical PFOS and PFOA could be causing reproductive effects, and if so, could the doses be realistically relevant?
Yeah, I think I'd have to go back to see whether any of those perfluorinated chemicals were a part of the taxonomical library. So I'd have to specifically look at those profiles. But for the concentration selection, I'm not sure how relevant those would be for human exposures. So I'd have to go back and specifically look at those profiles. But one thing that I didn't really mention is, so this work was recently published in Toxi, and all the supplemental data so all of the Mahalanobis distances that we've calculated, as well as all the steroid profiles, are available in the supplement. And so feel free to look at some of those. I know we had some questions about phthalate chemicals as well. So without specifically looking at having a profile in front of me and looking at those values, I can't really speculate too much. So
PFAS was, I think PFAS was definitely in there.
PFAS was in there?
Yeah.
Okay. Yeah, I'd have to double check, but that's something that I can definitely look into, and also feel free to browse the supplement yourself as well.
All right, great. I think we have time for one more question, if there's one in the room or on the chat box.
When you were looking at the, to try and choose like the distance that metric you used, did you look at anything like PCA or other methods that are specifically built for dimensionality reduction? Or I guess a better question is like, how did you end up on this as like your ending measurement?
Yeah. I think I'd want to probably defer to Woody on that one. But
so Mahalanobis distance was picked because of that correction for the covariance between the hormones that we know are interdependent in their synthesis. And so there's probably a lot of other metrics. One that's been mentioned to me is a two-keys-based metric that we could have used, but primarily, originally we were thinking about, distance, just the Euclidean distance, and we thought, hey, that's not appropriate. We need a distance metric that actually deals with the covariance between hormone measures in addition to the variance of a single hormone measure, right? So that's why we selected it, but obviously it was the simplest approach, sort of a derivative of Euclidean distance, but there's many other metrics that might be appropriate for that kind of data, as long as they're correcting for the variance for the hormone A itself, as well as the covariance to all the hormones in the set.
Okay.
yeah, I'm also trying to think of how PCA would factor in, because I think it would be different. It would depend on how we would analyze the data, where we would want to take a PCA based on all of the data overall, or if we would stick it within just within a chemical concentration response. So the Mahalanobis distance let us control for that covariance issue on the entire data set, and I'm not sure how PCA, like a PCA approach, would handle something like that, because
yeah, I mean it would certainly be different, which would be maybe interesting to look at. But you could take the whole thing and maybe just look at the first coefficient. We're just going to have most of your variance anyway.
Yeah.
So I don't know, just something that occurred to me when you were talking.
Yeah, no, that's a good question.
All right, thank you. Well, that's the end of our time. I apologize for our technical issues. Thank you, Derik, and thank you, everybody, for hanging with us. I hope you all have a happy holidays, and hopefully you'll join us in January.
The leader has disconnected. The conference will be terminated in 5 minutes.